Okay, so far we completed the synopsis for the chapter plant growth and development. Let's discuss some questions from this chapter. Yeah, so bolting is associated with physiological function of which hormone? Plant hormone, gibberellic acid, cytokinin, IAA, ABA. Okay, so gibberellic acid is responsible for increase in internode. Okay, so by using this gibberellic acid, we can convert that cabbage into tall plants. So one cabbage can be made into tall plant by applying gibberellic acid. So here internode is condensed, but here internode elongation takes place that made the plant to increase in its length. Okay, that process is called as bolting. Okay, this bolting could be achieved by applying gibberellic acid. Okay, so there are many types of gibberellic acids were there, but like gibberellic acid 1, 2, 3, 4, like this, many were there. Gibberellic acid 2, gibberellic acid 3, like that, many were there, but most preferable gibberellic acid is gibberellic acid 3. Okay, so as you know, the gibberellic acid was first isolated by a scientist called Kurosawa by observing a plant disease, that rice plant disease called as Bekane disease or foolish seedling disease. So what he observed, so normal rice seedlings were have the height like this, but one few plants were shows very tall nature. Okay, so he called it as a foolish seedling disease. Okay, in Japanese it is called as Bekane disease. So later he identified one of the fungi called a Gibberella fugicori. Okay, so Gibberella fugicori. Gibberella fugicori is responsible for this disease. Okay, so later Kurosawa isolated a chemical from this Gibberella and he named it as gibberellic acid. Okay, so this gibberellic acid shows this bolting. Okay, so cytokinins, they are cell dividing help onto the cytokinin. IAA indole, 3-acetic acid, it's axin. ABA, abscisic acid. So next question number 2. Phytohormone which inhibit flowering in several plants but stimulate flowering in pineapple. Okay, so ಬೇರೆ ಪ್ಲಾಂಟ್ಸ್ಗಳಲ್ಲಿ ಫ್ಲವರ್ ಇಂಡ್ಯೂಸ್ ಆಗದೆ ಇರೋ ಥರ ಮಾಡುತ್ತೆ, ಬಟ್ ಇನ್ ಕೇಸ್ ಆಫ್ ಪೈನಾಪಲ್ ಸಿಂಕ್ರೊನೈಸೇಷನ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಸಿಂಕ್ರೋನಿಂಗ್ ಫ್ಲವರ್ ಇಸ್ ಕುಡ್ ಬಿ ಪ್ರೊಡ್ಯೂಸ್ಡ್ ಬೈ ಅಪ್ಲಿಕೇಶನ್ ಆಫ್ ಇಥಿಲಿನ್. ಓಕೆ, ಯಾ, ಸೊ ಇಥಿಲಿನ್, ಸೊ 2,4-D ಇಟ್ಸ್ ಆಕ್ಸಿನ್, EBA is axin, GA3 it is gibberellic acid. Question number 3. Okay, so part of the plant body where abscisic acid is not synthesized but a cytokinin synthesized. So which part contains the where no abscisic acid is synthesized, and it was not required at all at roots. Okay, so roots ಅಲ್ಲಿ abscisic acid synthesis ಬೇಕಾಗಲ್ಲ, ಯಾಕಂದರೆ root is under the soil and no need of that dormancy and all for that root. Okay, so this abscisic acid can accumulate in leaves, flowers and seeds. Okay, so maximum they are accumulated in the seed that leads to dormancy. So the seeds which are kept in the kitchen or in the boxes, they are not yet germinated because of this abscisic acid concentration. So when abscisic acid is present, seed it shows the dormancy. That dormancy can be break by application of gibberellic acid. So AB and gibberellic acid are opposite to each other with their function with respect to seed dormancy. Abscisic acid induces the seed dormancy, whereas gibberellic acid break the dormancy. Okay, yeah. So next question. So which of the following phytohormones have similar effect on leaf senescence? So senescence ಅಂದರೆ ಏಜಿಂಗ್ ಆಫ್ ಲೀಫ್. ಏಜಿಂಗ್ ಆಫ್ ಲೀಫಿಗೆ ಯಾವುದು ಎರಡು ಹಾರ್ಮೋನ್ಗಳು ಈಕ್ವಲಿ ಕಾಂಟ್ರಿಬ್ಯೂಟ್ ಮಾಡುತ್ತೆ ಅಥವಾ ಎರಡೂ ಕೂಡ ಸೇಮ್ ಫಂಕ್ಷನ್ ಮಾಡುತ್ತೆ ಅಂದರೆ ಇಟ್ ಇಸ್ ಅಬ್ಸಿಸಿಕ್ ಆಸಿಡ್ ಆ್ಯಂಡ್ ಇಥಿಲಿನ್. ರಿಮೆಂಬರ್ ದಿಸ್ ಟ್ರೀಟೆಡ್ ಆಸ್ ಪ್ಲಾಂಟ್ ಗ್ರೋತ್ ರಿಟಾರ್ಡರ್ಸ್. ಓಕೆ, ರಿಟಾರ್ಡ್ ಮಾಡುತ್ತೆ ಪ್ಲಾಂಟ್ ಗ್ರೋತನ್ನು. ಸೊ ದೋಸ್ ಆರ್ ದ ಆನ್ಸರ್. ಸೊ ಅಬ್ಸಿಸಿಕ್ ಆಸಿಡ್ ಆ್ಯಂಡ್ ಇಥಿಲಿನ್ ಆರ್ involved in senescence. Okay, so this senescence can be reduced by application of cytokinin and even auxin. Auxin and cytokinin can prevent the senescence. Okay. growth inhibitor responsible for stomatal closure. Stomatal close ಮಾಡಲಿಕ್ಕೆ ಯಾವುದು ಹೆಲ್ಪ್ ಆಗುತ್ತೆ ಅಂದರೆ ಇಟ್ ಇಸ್ ಅಬ್ಸಿಸಿಕ್ ಆ್ಯಸಿಡ್. ಓಕೆ, ಅಬ್ಸಿಸಿಕ್ ಆ್ಯಸಿಡ್ is stimulate the closure of stomata. Okay, zeatin is a cytokinin. At the same time, that cytokinin helps in opening of stomata. Okay. Okay. So study the following statement and select the correct one. Auxin is transported from base to tip in a large quantities. So plant na tip na ತಗೊಂಡ್ರೆ ಬೇಸ್ ಇಂದ ಟಿಪ್ಪಿ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಆಗುತ್ತೆ ಅಂತ ಹೇಳ್ತಾ ಇದ್ದಾರೆ. ನೆವರ್ ಅಕರ್ಸ್. ಸೊ ಸೆಲ್ಸ್ ಆನ್ ದ ಡಾರ್ಕ್ ಸೈಡ್ ಎಕ್ಸ್ಪ್ಯಾಂಡ್ ಫಾರ್ದರ್ ದ್ಯಾನ್ ಆನ್ ದ ಲೈಟ್ ಸೈಡ್. ಓಕೆ, ಸೊ ಸೆಲ್ಸ್ ಆನ್ ದ ಡಾರ್ಕ್ ಸೈಡ್ ಎಕ್ಸ್ಪ್ಯಾಂಡ್, ನಾಟ್ ಎಕ್ಸ್ಪ್ಯಾಂಡ್, ದೇ ಆರ್ ಎಲಾಂಗೇಟೆಡ್, ನಾಟ್ ಎಕ್ಸ್ಪ್ಯಾಂಡ್. ಓಕೆ, ಸೊ ದೇ ಆರ್ ಎಲಾಂಗೇಟೆಡ್, ನಾಟ್ ಎಕ್ಸ್ಪ್ಯಾಂಡೆಡ್. Okay, so apical dominance can be removed by the decapitation. If you remove the tip, of course you can remove the apical dominance. Tropical movement in plants are under the control of gibberellins. No, it is under the control of auxin. Okay, so C is the right answer. So auxin shows that the phototropism. So which one of the following pairs is not correctly matched? Adenine derivatives, of course kinetin and zeatin are adenine derivatives, whereas carotenoid derivatives are abscisic acid. So you know the pigment carotenoids. Okay, so carotenoids derivative in the synthesis of abscisic acid, carotenoid pigment is involved, whereas the terpenes, terpenes are synthesized. Terpenes are going to synthesize the gibberellic acid. Okay, then indole compounds, they are indole butyric acid and IAA. So they are indole compounds. Okay, yeah. If you remember that auxins are synthesized. Auxins are synthesized from that amino acid called tryptophan. So tryptophan is required, and zinc is also required for the synthesis of amino acid auxin. Okay. Next question. So cytokinin promote abscission, influence water movement, inhibit protoplasmic streaming, help retain chlorophyll. Yeah. So cytokinin helps in retain the chlorophyll. It can act as an antagonistic to the function of abscisic acid and ethylene. Abscisic acid promote the degradation of chlorophyll, whereas cytokinin try to retain the chlorophyll, and even it helps the synthesis of chlorophyll. So this is the right answer. D is the right one. Okay. So next question. Abscisic acid promotes cell division. Never, do not promote cell division. Leaf fall, senescence and dormancy. So leaf fall ಆಗೋಕ್ಕೆ ಮತ್ತು senescence ಗೆ ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ. Shoot elongation, no. Shoot elongation ಅಂದರೆ either it is auxin or gibberellin. Then cell elongation and wall formation. No. Cell elongation and wall formation by auxin. So abscisic acid, senescence ಬರುತ್ತೆ. Senescence and dormancy. Okay. So next question. Question number 10. Okay. So a green plant bends towards the source of light when exposed to the light on only one side. Which of the following is the best explanation of the phenomenon? ಯಾಕೆ ಬೆಂಡ್ ಆಗುತ್ತೆ? Okay. So the apex first time are attracted by the light. Attached ಆಗಿದೆ? No. So the plant needs light for photosynthesis. That is no relationship with the requirement of light for photosynthesis and bending. Okay. Then auxin accumulates on the shaped side of induced great cell elongation. Yes, a cell elongation on the other side. Light induces length. No. So when you given the light from one source and all the side, if it is a dark, from only one source if you provide the light, what happens? Auxin start accumulate here and elongate the cell in one side. One side ಅಲ್ಲಿ ಮಾತ್ರ elongation ಆಗುತ್ತೆ, ಈ ಸೈಡ್ ಮುದ್ದಾದ ಇದು ಬೆಂಡ್ ಆಗುತ್ತೆ. It starts bending towards the light. That is the reason. Okay, so auxin accumulate in the darker side, that should be remembered. So in a normal plant what happens? So since light is present throughout side, ಎಲ್ಲ ಸೈಡ್ ಇರೋದ್ರಿಂದ auxin can accumulate in the middle, away from the light. So it helps in elongation of the cells in the middle. So that helps in growth of a tip. Okay. Yeah. So next question. So morphogenesis is controlled by an interaction between. So morphogenesis is controlled by the interaction between the two plant hormones. One hormone that induces shoot system and another hormone that induces root system. So okay, shoot system is induced by cytokinin and the root system is induced by auxin. Okay, so this is the right answer. So closure of stomata is brought about by, of course, it is through abscisic acid only. Abscisic acid helps in closure of stomata. Okay. So next question, question number 14. So hormone responsible for aging or senescence, obviously no direct question, it is ABA only. Okay. So next question. So photoperiodism is probably due to the synthesis of, so photoperiodism means it is a flower induction when exposed to light. Okay, so the hypothetical hormone is called florisin. So florisin is responsible for flowering, and it is synthesized in the leaf and migrated to shoot tip, where it converts the shoot tip into flower primordia. Okay, that later flower is going to be produced there. So the phytohormone that helps in germination of seed. So germination in the sense, by breaking the dormancy, it helps the plant to get germinate, that is gibberellin. So how the seed is get germinated with the help of abscisic acid? If you take a monocot fruit, it's like this. So if you remember the structure, it has the alluran layer. Okay, so when you soak the seed, the gibberellic acid, the abscisic acid is usually, it is more concentrate of abscisic acid is more here. When you soak the seed, gibberellic acid is produced. That gibberellic acid, okay, so that gibberellic acid degrade this abscisic acid, alluran layer protein na degrade ಮಾಡುತ್ತೆ. Okay, so to protein na degrade ಮಾಡಿ, that protein that are involved in breakdown into amino acid, that amino acid is involved in the metabolism. So that metabolism produces the enzyme. That enzyme helps in germination of the seed. Okay, so this is how the gibberellic acid helps in production of influence on seed germination. Okay, so it is gibberellin. So when the seeds start germinated, means abscisic acid concentration reduces and gibberellic acid concentration increases. Okay. So next question. So programmed cell death is scientifically termed as, so programmed cell death is called as apoptosis. Okay, so apoptosis plants ಗಳಲ್ಲಿ ಯಾವ ಟೈಮ್ ನಲ್ಲಿ ಆಗುತ್ತೆ ಅಂದರೆ, suppose in a plant leaf, let's consider these are the cells which are present there. If any one of the cell is infected with the virus, it sends the signal to neighboring cell that I am attacked by the virus. Okay, so to avoid the further spreading of this virus to neighboring cell, these cells are going to be killed themselves. ಇದನ್ನು ಪ್ರೋಗ್ರಾಮ್ಡ್ ಸೆಲ್ ಡೆತ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಅಂದರೆ ಈ ಪಾರ್ಟಲ್ಲಿ ವೈರಸ್ ಸ್ಪ್ರೆಡ್ ಆಗೋದನ್ನ ಈಗ ಅವಾಯ್ಡ್ ಮಾಡಿಸ್ಬಿಡುತ್ತೆ. Okay, that is apoptosis, called as programmed cell death. So it's a kind of a defense mechanism in the plants to overcome or to fight against the pathogens. Okay, so next question. So leaf abscission is caused by, abscisic acid. So abscisic acid, how it forms the helps in the falling of leaf antandre. So there is a connection between the connection at the junction of stem and leaf illidiyalla. So at this part first abscisic acid start accumulate there. Okay, so it block the entry of water first. Illi water ಮತ್ತೆ food conduction tissues na ಸ್ವಲ್ಪ block ಮಾಡುತ್ತೆ. Then it starts synthesize the abscisic acid. That abscisic acid start accumulate there and completely separate or cut the connection between the leaf and stem. So ಇಲ್ಲಿ brown color formation ಆಗಿರುತ್ತೆ. That is due to abscisic acid. Later this leaf is get detached from the plant. That leads to senescence. Okay, so that is how abscisic acid is functioned, okay, in leaf. Okay, let's move to the next question. So plasticity in plant growth means that plant roots are extensible, plant growth is depend on environment. Yeah, so plasticity ಅಂದರೆ environmental ಕಂಡೀಷನಿಗೆ ಕರೆಕ್ಟಾಗಿ ಪ್ಲಾಂಟಲ್ಲಿ ಆಗುವಂಥ ಚೇಂಜಸ್ಗಳು. ಓಕೆ, ಸೊ ಇದನ್ನು ಸಮ್ಟೈಮ್ಸ್ ಬೆಸ್ಟ್ ಎಕ್ಸಾಂಪಲ್ ಅಂದರೆ ದ ಕಂಡೀಷನ್ ಕಾಲ್ಡ್ heterophyly is best explained by this plasticity. So heterophyly is a condition where plant can produce two kinds of leaves. Okay, so according to environmental condition. ಓಕೆ, ಸೊ ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಹೆಟೆರೋಫಿಲಿ. ಅಂದರೆ ಒಂದೇ ಪ್ಲಾಂಟಲ್ಲಿ 2 ಟೈಪ್ಸ್ ಆಫ್ ಲೀವ್ಸ್ ಇರುತ್ತೆ. ಈ ಥರದ್ದು ಲೀವ್ಸು, ಹೈಲಿ ಡಿಸೆಕ್ಟೆಡ್ ಲೀಫ್. These are the 2 kinds of leaves produced in single plant called as heterophily. ಅದು ಎನ್ವೈರ್ಮೆಂಟಲ್ ಕಂಡೀಷನ್ಗೆ ಅಡಾಪ್ಟ್ ಆಗಿ ಪ್ಲಾಂಟಲ್ಲಿ ಆಗುವಂಥ ಚೇಂಜಸ್ಗಳು. ಓಕೆ, ಸೊ ದಟ್ ಈಸ್ ಪ್ಲಾಸ್ಟಿಸಿಟಿ. ಯಾ, ದ ನೆಕ್ಸ್ಟ್ ಕ್ವೆಶನ್. ಸೊ ಕ್ವೆಶನ್ ನಂಬರ್ 20. Okay, so which of the following statement is not related with the given curve? LT equals L0 plus RT, L0 plus RT. So sigmoid growth curve, geometric growth, typical for cells in culture and many higher plants and plant organs. Okay, so whatever the thing they are shown is a sigmoid curve. So it has a lag phase, a log phase and a stationary phase. So for this the formula is W2 equals W0 plus RT. So this equation is for geometric growth. This is for geometric growth, whereas this for arithmetic growth. Okay, it is for arithmetic growth. Okay, so arithmetic growth ಅಂದರೆ linear ಇರುತ್ತೆ. So linear equation ಇದು. Okay, it is not for this because this is a geometric progression. So geometric progression observed in cell in culture and some bacteria. Okay, ಅಂಥ ಕಡೆಯಲ್ಲಿ ಮಾತ್ರ ಇರುತ್ತೆ. ಸೊ ದಿಸ್ ಇಸ್ ನಾಟ್ ಹೋಲ್ಡ್ಸ್ ಗುಡ್ ಫಾರ್ ದಿಸ್ ಡಯಾಗ್ರಾಮ್. ಓಕೆ, ಸೊ ಇದು ಸಿಗ್ಮಾಯ್ಡ್ ಕರ್ವ್ ಹೌದು, ಜಾಮೆಟ್ರಿಕ್ ಗ್ರೋತ್ ಹೌದು. ಇಟ್ ಇಸ್ ಅಬ್ಸರ್ವ್ಡ್ ಇನ್ ಸೆಲ್ ಕಲ್ಚರ್ ಆ್ಯಂಡ್ ಮೆನಿ ಹೈಯರ್ ಪ್ಲಾಂಟ್ಸ್ ಆ್ಯಂಡ್ ಪ್ಲಾಂಟ್ ಆರ್ಗನ್ಸ್. ಸೊ ಪ್ಲಾಂಟ್ ಆರ್ಗನ್ಸ್ಗಳಲ್ಲಿ ಆಗ್ತಾ ಇರುತ್ತೆ. ಇಫ್ ದ ಸೆಲ್ ನಂಬರ್ ನೀಡ್ ಟು ಬಿ ಇನ್ಕ್ರೀಸ್ಡ್, ಆ ಕೇಸಲ್ಲಿ ಮಾತ್ರ ಆಗುತ್ತೆ. ಸೊ ಫಾರ್ ಇಫ್ ಯು ಕನ್ಸಿಡರ್ ಫಾರ್ ಎ ಗ್ರೋತ್ ರೇಟ್, ಅರ್ಥಮೆಟಿಕ್ಗೆ ಈ ಈಕ್ವೇಷನ್ ಯೂಸ್ ಮಾಡ್ತೇವೆ. LT equals L0 plus RT. Okay, so T is the time, L0 is the initial and RT is the rate of cell division, then T is the time. Okay, so let's go to the next question. Question number 21. Yeah, so match the following with plant growth regulators and their precursor. ಅದು ಯಾವುದರಿಂದ ಸಿಂಥಸೈಸ್ ಆಗುತ್ತೆ ಅನ್ನೋ ಬೇಸಿಸ್ ಮೇಲೆ we have to identify it. Okay, yeah. Kinetin, AVA, G and C2H4. So kinetin is adenine derivative. Kinetin is a adenine derivative. Remember that a Skoog and co-worker, when they worked on tobacco plant pith culture, so they are unable to get the callus properly. So accidentally they add that adenine derivative, which is isolated from herring sperm of DNA. Okay, herring fish the DNA isolate ಮಾಡಿರ್ತಾರೆ, ಅದರಿಂದ ಹಾಕಿರ್ತಾರೆ. ಸೊ ಇಟ್ ಪ್ರೊಲಿಫರೇಟ್ ದ ಸೆಲ್ ಡಿವಿಷನ್. ಓಕೆ, ಸೊ ಹಾಗಾಗಿ ಆಡ್ನಿಂದ ಸೈಟೋಕೈನಿನ್ ವಾಸ್ ಡಿಸ್ಕವರ್ಡ್. ಓಕೆ, ಸೊ ಥರ್ಡ್ ಒನ್ ಆಯಿತು. ದೆನ್ ಅಬ್ಸಿಸಿಕ್ ಆ್ಯಸಿಡ್. ಸೊ abscisic acid is a carotenoid derivative. You know the pigment xanthophyll A, chlorophyll A, chlorophyll B, xanthophyll and carotenoid. So that is a pigment, that yellow color pigment that is responsible for precursor for ABA. A carotenoid indane abscisic acid synthesize agake start agutte. Okay, and gibberellic acid. So these are derivatives of terpenes. Okay, so terpenoids group ಇದೆ ಪ್ಲಾಂಟ್ಸ್ ಗಳಲ್ಲಿ. ಆ ಟರ್ಪಿನ್ದು ಒಂದು ಅದರಲ್ಲಿ ಮಾನೋಟರ್ಪಿನ್, ಡೈಟರ್ಪಿನ್, ಟ್ರೈಟರ್ಪಿನ್, ಈ ಥರ ಮೆನಿ ಟೈಪ್ಸ್ಗಳಿದೆ. ಸೊ ದಿಸ್ ಈಸ್ ನಥಿಂಗ್ ಬಟ್ ಇಥಿಲಿನ್. So 1 3 1 3 1 3. So you can eliminate this one, then 2 4. So 2 4. So you can directly eliminate this, you can choose it. Okay, yeah. Next question number 22. Okay, so question number 22. What would be expected to happen if you cut the shoot tips of tea plant na shoot tip na cut ಮಾಡಿದ್ರೆ ಏನಾಗುತ್ತೆ? ಥರ ಇರುತ್ತೆ. ಸೊ ಈ ರೀತಿ ಶೂಟ್ ಟಿಪ್ನ ರಿಮೂವ್ ಮಾಡಿದ್ರೆ ಏನಾಗುತ್ತೆ? ಸೊ ಇನಿಬಿಷನ್ ಆಫ್ ಲ್ಯಾಟ್ರಲ್ ಗ್ರೋತ್. ದೀಸ್ ಆರ್ ಲ್ಯಾಟ್ರಲ್ ಬಡ್ಸ್, ಆಕ್ಸಿಲರಿ ಬಡ್ಸ್. So growth of lateral buds, promotion of horizontal growth of male flower, inhibition of flower. Okay, so idina remove madidre apical dominance is removed. So once apical dominance is removed, branches starts to produce there, that make the plant bushy. Okay, so it is a growth of lateral buds. So apical dominance is for auxin. Question number 23. So choose the correct option on the basis of a comparison between the size of two leaves A and B given below, so leaf A and B. So A initial size is 5 centimeter. Initial size is 5 centimeter. Final size is 10 centimeter. Whereas in B, initial is 50 centimeter and final is 55 centimeter. Okay, so leaf A shows the less relative rate of growth, leaf B shows the less absolute growth, leaf B shows more relative rate of growth, both leaves show same absolute growth. Okay, so absolute growth 5 ಇದ್ದಿದ್ದು ಎಷ್ಟು ಜಾಸ್ತಿ ಆಗಿದೆ 5 centimeter. So in case of A, absolute growth, this absolute growth is only 5 centimeter. So 5 to 10 increase ಆಗಿದೆ. So difference ಎಷ್ಟು 5 centimeter ಅಷ್ಟೇ. So 5 centimeter is the actual growth. Here in this case, final stage is 50 ಆಗಿದೆ. So here also same, absolute growth is the same in both the case. Okay, if you calculate a relative growth, so relative growth ಅನ್ನು ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡಬೇಕಂದ್ರೆ ಫೈನಲ್ ಗ್ರೋತ್, ಫೈನಲ್ ಸೈಜ್ ಮೈನಸ್ ಇನಿಷಿಯಲ್ ಸೈಜ್ ಡಿವೈಡೆಡ್ ಬೈ ಇನಿಷಿಯಲ್ ಸೈಜ್ ಇಂಟು 100. ಓಕೆ, ಸೊ ಫಾರ್ ದ ಫಸ್ಟ್ ಕೇಸ್ ಎ ಕೇಸಲ್ಲಿ ತೊಗೊಂಡ್ರೆ ಫೈನಲ್ ಎಷ್ಟಿದೆ 10 ಇದೆ, ಇನಿಷಿಯಲ್ 5 ಇದೆ, ಇನಿಷಿಯಲ್ 5 ಇದೆ ಇಂಟು 100. ಓಕೆ, ಸೊ ಇಟ್ ಈಸ್ 5/5 ಇಂಟು 100. ಸೊ ಇಟ್ ಈಸ್ 100%. ಸೊ 100% ಗ್ರೋತ್ ಆಯಿತು. ಇಫ್ ಯು ಅಪ್ಲೈ ದ ಸೇಮ್ ಥಿಂಗ್ ಫಾರ್ ಲೀಫ್ ಬಿ, initial final is 50, initial is final is 55, initial is 50 divided by 50 into 100, then it is 5/50 into 100. Okay, 5 ones are 5 twos are, so it is only 10%. Okay, so 10%. So absolute growth is same but relative difference. Okay, both leaves show same absolute growth. Yeah, both shows the same absolute growth. ಎರಡು ಕೂಡ same absolute growth show ಮಾಡ್ತಾ ಇದೆ. So this is the answer. Yeah, so hope you got it. Next question number 24. Yeah. Certain plants also need to be exposed to low temperature so as to hasten the flowering. This treatment is known as, not only for flowering, this process is applicable for seed germination also, that is called vernalization. So vernalization is the exposing of a seed or a seed for germination and a plant for low temperature treatment to induce the flowering, called as vernalization. Okay, so it is not applicable to all the plants. Few plants require this vernalization. Okay. Okay. So find the correct match with respect to growth regulators. Promote the abscission of older mature leaves and fruits. Okay. So ಯಾವುದು promote ಮಾಡುತ್ತೆ? Okay. So 1, 2, 3, 4. Promote the abscission of older and mature leaves. Okay. Increase the length of grape stalk. So direct ಆಗಿದೆ. You can choose this. 3(1), B3. Then promote nutrient mobilization. So nutrient mobilization, promote a root growth and root hair formation. Root hair formation is by ethylene. Sorry, promote root growth. Promoting root growth means this one. Second one, D is 2. D is 2. So D is 2 illide. Okay, so they can eliminate remaining. Okay, so ನೋಡೋಣ A4. Promote the abscission of older mature leaves and fruits. Yeah, even this auxin do this process. Okay, auxin promote the abscission of older leaves but prevent the abscission of younger leaves. Okay, B3 increase in the length grape stalk, that is gibberellin. Okay, so B3 gibberellin. Then C1 promote the nutrients mobilization, it is ethylene. Then ethylene. Then D2, that is promote a root growth and the root formation, is cytokinin. But here one point I need to point out. So ethylene helps in root hair formation. It helps in root hair formation. Okay, similarly auxin helps in root formation. Okay, so root hair formation help ಮಾಡುತ್ತೆ. Okay, yeah. So in that sense promote nutrient mobilization. Nutrient na ಜಾಸ್ತಿ ಅಬ್ಸರ್ವ್ ಮಾಡ್ಕೊಳ್ಳೋಕ್ಕೆ ರೂಟ್ ಹೇರ್ ಜಾಸ್ತಿ ಇದ್ದಷ್ಟು more nutrients is get absorbed. ಹಾಗಾಗಿ ethylene can promote this function. Question number 26. Yeah, question number 26. chilling treatment in plant is required for, that is also called as vernalization, inducing flowering, promotion of flowering, germination of seeds. So more than one option is correct. Okay, so D is the right answer. Okay, especially the process is required for some wheat plant and some temperate countries plant, they need this chilling treatment usually. Okay, next question. So auxin was isolated by dash from tips of coleoptile of which plant? Okay, so it is called a bioassay. ಆ ಬಯೋಅಸ್ಸೇನ ಮಾಡಿದಂಥವ್ರು ಯಾರು, ಅದನ್ನು ಆಕ್ಸಿನ್ ಇದೆ ಅಂತ ಪ್ರೂವ್ ಮಾಡಿದವ್ರು ಯಾರು ಅಂದರೆ ದರ್ ಇಸ್ ಅ ವೆಂಟ್, ಸೈಂಟಿಸ್ಟ್ ಕಾಲ್ಡ್ ವೆಂಟ್ ಇನ್ ವೋಟ್ ಪ್ಲಾಂಟ್. ಬಟ್ ಈ ಕೆನರಿ ಗ್ರಾಸಲ್ಲಿ ಡಾರ್ವಿನ್ ಮಗ ಇದ್ರಲ್ಲ, ಇವರು ಸ್ಟಾರ್ಟ್ ಮಾಡಿದ್ರು ಫೋಟೋ ಪೀರಿಯಾಡಿಸಮ್ ವರ್ಕ್ ರಿಲೇಟೆಡ್ ಈ ಬಿಗಿನ್ಸ್. ಸೊ ಫೈನಲಿ ಅದನ್ನು ಆಕ್ಸಿನ್ ಅಂತ ಐಸೋಲೇಟ್ ಮಾಡಿ ಇದು ಮಾಡಿದವರು ವೆಂಟ್ ಅಂತೇಳಿ, ಸೈಂಟಿಸ್ಟ್ ನೇಮ್ ಇಸ್ ವೆಂಟ್. ಓಕೆ, ಸೊ ಇದನ್ನು ಅವೈನಾ ಕರ್ವೇಚರ್ ಟೆಸ್ಟ್, ಕೆನರಿ ಗ್ರಾಸ್ನ ಅವೈನಾ ಅಂತೀವಿ, ಅವೈನಾ ಕರ್ವೇಚರ್ ಟೆಸ್ಟ್ ಎಲ್ಲ ಪ್ರಿಡಿಕ್ಟ್ ಮಾಡ್ತಾರೆ. ಓಕೆ, ಸೊ next question, number 28. So 2,4-D stands for 2,4-dichlorophenoxyacetic acid. That's 2-chlorophenoxy acetic acid. So this is the short form is 2,4-D. So remember 2,4-D is a synthetic one. It is a synthetic axin. Artificially synthesis ಮಾಡಬಹುದು, and it can kill the dicot weeds in lawn. ಲಾನಲ್ಲಿ ಡೈಕಾಟ್ ಪ್ಲಾಂಟ್ಸನ್ನು ಕಿಲ್ ಮಾಡೋಕ್ಕೆ 2,4-D ಸ್ಪ್ರೇ ಮಾಡ್ತಾರೆ. ಬಟ್ ದ ಎಕ್ಸಾಕ್ಟ್ ಫಿನಾಮಿನನ್ ಬಿಹೈಂಡ್ ಕಿಲ್ಲಿಂಗ್ ಓನ್ಲಿ ದಟ್ ಡೈಕಾಟ್ ಪ್ಲಾಂಟ್ ಇಸ್ ನಾಟ್ ನೋನ್ ಟಿಲ್. ಓಕೆ, ಇನ್ನೂ ಕರೆಕ್ಟಾಗಿ ಗೊತ್ತಿಲ್ಲ ಯಾಕೆ ಕಿಲ್ ಮಾಡುತ್ತೆ ಅಂತ ಹೇಳಿ. ಓಕೆ, ಬಟ್ ಇಟ್ ವಿಲ್ ನಾಟ್ ಹಾರ್ಮ್ ಮೊನೊಕಾಟ್, ಓನ್ಲಿ ಕಿಲ್ ಡೈಕಾಟ್ ಪ್ಲಾಂಟ್ಸ್. ಓಕೆ, ಸೊ ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್. So study the following list. Xylem in primary body, interfascicular cambium, phloem in the woody organ, cork cambium. So product of differentiation, so de-differentiation, product of differentiation, undergoes differentiation, product of de-differentiation. Okay, so first is xylem in a primary body. So xylem is a primary body ಅಂದರೆ just plant germinate ಆಗಿ ಅದು ಎಂಬ್ರಿಯೋ ಇಂದ ಪ್ಲಾಂಟ್ಲೆಟ್ ಬಂದಾಗ xylem ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ. ಇಟ್ ಇಸ್ ಅ ಪ್ರೈಮರಿ ಪ್ರೋಸೆಸ್. ಓಕೆ, ಸೊ ಅಲ್ಲಿ ಆಗುವಂಥ ಪ್ರೋಸೆಸ್ಸು xylem ಫಾರ್ಮೇಷನ್ ಆಸ್ ಅ ಪ್ರೈಮರಿ xylem ಫಾರ್ಮೇಷನ್ is an example for differentiation. Okay, so it's an example for differentiation. So next, interfascicular cambium. So interfascicular cambium, if you imagine the interfascicular cambium is the cambium which is produced in between the two vascular bundles. So ಇಲ್ಲಿ parenchyma cells ಇರುತ್ತೆ. These parenchyma are already matured one. They regain the ability of division called as a differentiation. So during differentiation it become meristematic. Now this cell undergo division to form mini cells. So these cells again converted into xylem or phloem. Okay, ಆದರೆ ಇಲ್ಲಿ interfascicular cambium ಏನಿದೆ, that is the product of de-differentiation. Okay, interfascicular cambium is formed in between the vascular bundles because of de-differentiation. So interfascicular cambium is the product of de-differentiation. Yeah, so B4. Okay, so let me check A2 B4. A2 ಎಲ್ಲದರಲ್ಲೂ ಇದೆ, B4, so we can eliminate this. Okay, so only 2 over there. Then phloem in the woody organ, then a cork cambium. Okay, so cork cambium option, sorry ಈ ಕಡೆ ಬಂದುಬಿಟ್ಟಿದೆ, so it should be here, product of redifferentiation. Okay, so ಇದು 5th option ಇಲ್ಲಿ ಬರಬೇಕು. Okay, so see phloem in the woody organ. Okay, so phloem during secondary growth, that cambium is the product of de-differentiated product. This undergo division to form few cells inside that become secondary xylem, the cell that become secondary phloem. The formation of this secondary phloem is redifferentiation. So redifferentiation, then C should be 5. So C should be 5. Okay, ಇಷ್ಟ ಆಯಿತು. Okay, then come to the last one, cork cambium. So cork cambium is the product of de-differentiation. Yeah, it is de-differentiated product. Okay, so A is the right one. Yes, so hope you got it. So ಸ್ವಲ್ಪ ಯೋಚನೆ ಮಾಡಿ ಆನ್ಸರ್ ಮಾಡಬೇಕಾಗತ್ತೆ. ಈ ಥರದ್ದನ್ನು ಹರಿಬರಿ ಮಾಡ್ಕೋಬೇಡಿ, ನಿಧಾನಕ್ಕೆ ಯೋಚನೆ ಮಾಡಿ ಹಾಕಿ, ಗೊತ್ತಿದ್ದು ತಪ್ಪಾಗುತ್ತೆ. ಓಕೆ, ಸೊ ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್ ನಂಬರ್ 30. Yeah. So Darwin, Wendt, Kurosawa, Skugan Miller, ಏನೇನು ಕಾಂಟ್ರಿಬ್ಯೂಷನ್ ಅಂತೇಳಿ. ಸೊ ಡಾರ್ವಿನ್ ಅಂಡ್ ಕ್ರಿಸ್ಟಲೈಸೇಷನ್ ಆಫ್ ಕೈನೆಟಿನ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ಸ್ ವಿತ್ ಕೋಲಿಯಾಪ್ಟೈಲ್ ಆಫ್ ಕ್ಯಾನರಿ ಗ್ರಾಸ್, ಗೊತ್ತಿದೆ ನಿಮಗೆ. A2 A2 3ರಲ್ಲಿದೆ, ಇನ್ನೊಂದು 4ರಲ್ಲಿದೆ. ಓಕೆ, ಸೊ ವೆಂಟ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ವಿತ್ ಕೋಲಿಯಪ್ಟರ್ ಕ್ಯಾರಿಯರ್ಸ್ ಕೈನೆಟಿನ್, ಐಸೋಲೇಷನ್ ಆಫ್ ಗೇಷಿಯಸ್ ವೈಟ್ ಹಾರ್ಮೋನ್, ಐಸೋಲೇಷನ್ ಆಫ್ ಆಕ್ಸಿನ್, ಬಿಕಾನಿಸಿನ್. ಓಕೆ, ಸೊ ವೆಂಟ್ ಐಸೋಲೇಷನ್ ಆಫ್ ಆಕ್ಸಿನ್. Okay, so B4, so you can eliminate this too. Okay, next Kurosawa, isolation of Bicane disease, so C5 ಎರಡರಲ್ಲೂ ಇದೆ. then ಇನ್ನೊಂದು ಯಾವುದು ಉಳ್ಕೊಳ್ತು, D. Okay, so Skoog-Miller kinetic. Okay, so D1, so it is D1, so this is the right answer. Then you should eliminate this. So B is the right one. Okay, let's go to the next question. Question number 31. Okay, so the rosette habit of cabbage can be changed by application of, it can be made into a larger tree by application of gibberellic acid. So direct question, it is gibberellic acid. Okay, so next move to the next question, question number 32. Okay, question number 32. Hormone primarily connected with the cell division. Yeah, direct question. It is either cytokinin or zeatin. Okay, so zeatin is a type of cytokinin. Okay, so next. So the caseous fruit ripening phytohormone, direct question. It is ethylene. Ethylene is responsible for ripening. Okay. So next question 34. Yeah, so gibberellins were first extracted from coleoptile tip, root tip, fungus and bacteria. It is isolated from fungus, Gibberella fugicori by Kurosawa. Okay, he observed the foolish seedling disease in rice plant. So based on that he isolated the chemical from fungi, that fungi is responsible for that foolish seedling disease. So from fungi he isolated a chemical. When he spray that chemical again to the healthy plant, it will become tall. So that proves that fungus is responsible for the production of that particular chemical. Okay, so that chemical later find out in all other plants as a plant growth regulator. Okay, next question number 35. Growth promoter hormones. So growth promoter andre auxin, gibberellin, cytokinin. Okay, so auxin, gibberellin, cytokinin I have to identify. Auxin, so ABA is there, so it is not the option. ABA is there, it is not the option. ABA is there, it is not the option. So this is the option. IAA, indole 3 acetic acid, gibberellic acid and cytokinin. So C is the right answer. Okay, next question. Question number 36. Yeah, question number 36. So current sequence of different phases of growth. First cell division, cell differentiation, cell angle. No, first ಹೇಳ್ಬಿಡ್ತೀನಿ. ಫಸ್ಟ್ ಏನಾಗುತ್ತೆ, ಸೆಲ್ ಡಿವಿಷನ್ ಆಗುತ್ತೆ. ಸೆಲ್ ಡಿವಿಷನ್ ಆಗಿ 2 ಸೆಲ್ ಆಗುತ್ತೆ. 2 ಸೆಲ್ ಇದ್ದಿದ್ದು ಎಲಾಂಗೇಷನ್ ಆಗುತ್ತೆ. ಎಲಾಂಗೇಷನ್ ಆದಮೇಲೆ ಇಟ್ ಗೆಟ್ ಡಿಫರೆನ್ಷಿಯೇಟೆಡ್, ಮೆಚ್ಯೂರ್ ಆಗುತ್ತೆ. ಸೊ ಫಸ್ಟು ಸೆಲ್ ಡಿವಿಷನ್ ಇರಬೇಕು. ಸೆಲ್ ಡಿವಿಷನ್, ಓಕೆ. ಸೆಲ್ ಡಿಫ್ರೆನ್ಷಿಯೇಷನ್ ನೋ, ಸೆಲ್ ಎಲಾಂಗೇಷನ್ ನೋ, ಇವೆರಡು ಅಲ್ಲ, ಇವೆರಡರಲ್ಲಿರುತ್ತೆ ಸೆಲ್ ಡಿವಿಷನ್. ಸೊ ನೆಕ್ಸ್ಟ್ ಪ್ರೊಸೆಸ್ ಶುಡ್ ಬಿ ಸೆಲ್ ಎಲಾಂಗೇಷನ್. ಇಲ್ಲಿ ಡಿಫ್ರೆನ್ಸಿಯೇಷನ್ ಇದೆ, ಸೊ ಇಟ್ ಇಸ್ ನಾಟ್ ದ ರೈಟ್ ಒನ್. ಸೆಲ್ ಎಲಾಂಗೇಷನ್, ಸೆಲ್ ಡಿಫ್ರೆನ್ಸಿಯೇಷನ್. ಸೊ ಡಿ ಈಸ್ ದ ರೈಟ್ ಆನ್ಸರ್. Okay, go to the next question. Question number 37. Yeah. So which of the following cannot be taken as a parameter for measuring the growth of a plant body? Plant body growth and measure ಮಾಡಕ್ಕೆ ಯಾವ್ದು ಕನ್ಸಿಡರ್ ಮಾಡೋಕ್ಕಾಗಲ್ಲ. Fresh or dry weight, increase in biomass can be considered. Thickness, increase in the thickness of a leaf lamina. So leaf lamina thick ಆಗ್ತಾ ಇದೆ ಅಂದರೆ it is not considered as a growth. It is simply accumulation of organic material there. Then surface area of leaf lamina, yes, you can consider. Then the length of a root stem, yes, we can consider. So B is the right option. So thickness of leaf is not a measurement for growth. Okay, so next question. Question number 38. So heterophilous aquatic plant of the following is cotton, coriander, larkspur, buttercup. Yeah, so it is buttercup. So buttercup shows the heterophily. 2 types of leaves it shows. One is helps in the under the water, another one floating on the water. So buttercup is the right answer. Okay, even coriander also shows the heterophily, but they are asking for aquatic plant, whereas larkspur and coriander are for land plant, but buttercup is for aquatic, hydrophytic plant. Okay, so next question. So which of the following is a naturally occurring oxygen? Indole 3 butyric acid, naphthalene acetic acid, 2,4,5 trichlorophenols, acetic acid, 2,4-D. Well, synthetic oxygen. So Okay, so NAA, but IBA is the actual one. Okay, so naturally occurring is IBA as well as IAA, indole 3 acetic acid and butyric acid, eradu kuda naturally occurring. Okay, so next question. Select the incorrect statement of the following. If suspension of growth is due to the endogenously controlled factors, then it is termed as dormancy. Okay, it is a suspension of growth is due to endogenously controlled factors, then it is termed as dormancy. Okay, so of course we can consider dormancy. So olagadene embryonic growth agidru seed not germinated, so that's also due to dormancy. Criscograph used to measure RQ. No, criscograph is used to measure the growth. Okay, not RQ. Growth in plant is both determinate and indeterminate. ಎರಡು ಥರದ್ದು ಇರುತ್ತೆ ಪ್ಲಾಂಟ್ಸಲ್ಲಿ. ಗ್ರೋತ್ ಇನ್ ಪ್ಲಾಂಟ್ ಈಸ್ ಲೋಕಲೈಸ್ಡ್. ಎಸ್, ಇಟ್ ಈಸ್ ಲೋಕಲೈಸ್ಡ್. ಓಕೆ, ಸೊ ಬಿ ಇಸ್ ದ ರೈಟ್ ಆನ್ಸರ್. ಕ್ರಿಸ್ಕೋಗ್ರಾಫ್ನ ಗ್ರೋತ್ ಮೆಷರ್ ಮಾಡಲಿಕ್ಕೆ ಯೂಸ್ ಮಾಡ್ತೇವೆ. ಓಕೆ, ಸೊ ದಟ್ಸ್ ಇಟ್. ದಿಸ್ ಕಂಪ್ಲೀಟ್ಸ್ ದ ಚಾಪ್ಟರ್ ಕ್ವೆಶನ್ಸ್. ಥ್ಯಾಂಕ್ ಯು.